take it away, Lisa. Tell them about yourself, please.
Thank you. Well, there isn't a lot to say. I work mostly in the environmental arena. I kind of cover a lot of bases just because I'm interested in a lot of different things. So work a little bit in the hard sciences, a little bit in the soft sciences, some outreach work. Um, I it's funny because a lot of people who know me from one place are very confused when they hear my name pop up in a whole other area because they don't realize that I have a lot of interests. But I think so. I'm not going to talk a lot about myself. I'm happy to, if later on, if people have specific questions, I think as I go along, you'll learn about me a little bit. Um, and and hopefully learn about genetics and hereditary traits and inheritance. That's what we're here for today. So this can be a pretty deep subject. I tried to take a middle line here because I don't want people to be bored out of their wits, because I know this audience knows a lot about plant genetics already, probably, but maybe some of us are rusty. So hopefully it's a little bit of a refresher, and hopefully there are at least one or two things that you learn if you know a lot already. And if not, hopefully I'm not going to go too in depth. So I'm going to start. And I want to start with this because all of us here, unless you're out of state or not in San Diego, not in this area, we're on Kumeyaay land or Luiseno or Quechuan. And so this is my way of giving a land acknowledgement. I really like this quote: all plants are our brothers and sisters. They talk to us, and if we listen, we can hear them. This is a good reminder that genetics and inheritance and everything that I'm going to be talking about today, this kind of plant breeding and caring for plants and stewardship was being done for millennia. And maybe they didn't have the tools to understand chromosomes and things like that. But if it wasn't for the great stewardship of all of our tribal and indigenous people around the world, we might not have plants or as many as we have right now. So that's my thank you. So today's topics, I'm going to talk about three things basically. One is plant genetics, and we're going to be focusing on angiosperms specifically. Then we're going to talk a little bit about intraspecies hybridization, so not talking about crossing between different species, both the kind of the history of it. We're going to be talking about Mendel and his famous pea plants, and then just touch a little bit on kind of the difference between what was known then and what's known now that we have all kinds of technology that didn't exist in the past. And then I'm going to talk briefly a little bit about Ranunculus asiaticus. For 7 years, I worked for Milano and Company and a plant breeding on their breeding program. I worked with Ruth Kobayashi. I'm sure a lot of you know who she is. And so I'm going to talk for a few minutes about what we did there and what is still going on, because that program is still live. So what are angiosperms? And again, I don't mean to be demeaning, but sometimes we're like, wait a minute, what was that? So we're just going to cover this quickly just to make sure no one's left behind. So they're flowering plants, and they specifically anything that has fruit or flowers, they're angiosperms, and we'll get to the alternative in a second. This was a great evolutionary change that occurred. and it makes for tasty treats for birds and pollinators, and it makes for tasty treats for whoever wants to eat these things. And it adds this ability to transport genetic material. So really at the heart of everything today, the idea is that this is genetic material. You're trying to keep your species alive. So everything that we're learning is about how organisms have tried to do that. So angiosperms make up 80% of land plants, and there are over 300,000 species that have been identified so far. So the other plants, the other seed plants that we're not going to be talking about are gymnosperms, and they just consist of these four groups: conifers, cycads, neophytes, and ginkgo. They dominated the landscape throughout the Cretaceous period. But they had their day clearly, because now there are only about 1,000 species. And so as I was saying in the last slide, you can see how through evolution created this method to transport seeds in a way that would be more efficient and effective over just having the naked seed, which is what gymnosperm means. And so obviously that worked, because now there are so many of the angiosperms compared to gymnosperms. So very quickly, as you all know, to us as humans, angiosperms are really important for horticulture and gardening. They make up the majority of plants in landscaping and ornamental gardening and crop production. They exist in diverse forms and colors and have all kinds of different reproductive structures. That's what we're looking at when we're looking at these flowers. Again, what they're trying to do is pass on their genetic material. They're trying to look really attractive to whoever they're trying to get to help get their pollen somewhere else. So this is just a fantastic array of examples right here of what Mother Nature has done and what evolution has done over time to create this diversity. And so the purposes for humans: food production, obviously, all of these, aesthetic, ecological restoration, those are usually the purposes that are seen. Also, conservation, if you think about people buying certain plants to attract certain pollinators, right? Like with monarchs and things like that. So we actually do we practice conservation through our horticulture and gardening practices. So now we're going to start kind of a slow dip into into what we're going to get into, and just talk about the basics of plant genetics. Well, genetics in general, it's the study of how traits are inherited, expressed, and modified in plants. So how traits being like your eye color, or hair color, your height, all of those things. So not just how they're inherited, but how they're expressed, because there can be certain environmental factors and things like that cause the expression to be different. And then modified, same thing. It could with plants, as we know, and also with animals, you can have genetically modified organisms. And when you think about it, everything is genetically modified. Everything that we see through evolution is a form of genetic modification over time. But then GMOs, of course, are modification done by humans. And then we're also, when you're talking about plant genetics, you're looking at the genetics of individuals, and then you're also looking at what happens going from generation to generation and how that can affect a population. Again, back to the whole evolution thought, there could end up being maybe even a genetic mutation or something like that in one individual that proves to be advantageous, and it can change a whole plant population. This happens all the time. Again, that's the genetic modification. So we can't talk about genetics and inheritance without talking about Mendel. Johann Gregor Mendel, I don't speak or read or know any German. Maybe some of you can read this, but this was at the cover of the front of the book that he published, the famous book that depicted the 30,000, nearly 30,000 experimental pea plants or pea plant experiments that he did. And what he was doing was looking at patterns of transmission, trying to see if he crossed one thing with another, if what would happen in the next generation. Again, the individual versus population, he knew what the individuals looked like. They had yellow peas, they had, they were dwarf or they were high or, tall. So he knew the individual and what the individual traits were, and he liked them, or he, he was trying to see what would happen if he paired them up with something else and what would happen in the next generation. So he was looking for those patterns. And so for any experiments that you're going to do, as you know, there are the famous tsetse fly, or sorry, not tsetse fly, fruit fly experiments that are always done. And it's for these same reasons that you see that Mendel found pea plants to be model organisms. One is that they have a really short generation time. Could you imagine if he tried to experiment on a big, huge tree or something like that? Then we wouldn't, he wouldn't have been able to give us any of this knowledge that we have. Somebody else would have, though. He could control the breeding really easily. It was really easy to do pollinations, self-pollinations, cross-pollinations. And so that's really important. He had the tools were there. He didn't need any, technology that didn't exist at the time. There's variability. There were several different traits he could look at, and actually see differences as he went from one generation, across the parents from one generation to another. And there are a lot of offspring. In order to conduct really good experiments, you have to be able to statistically analyze the data that you're getting. And if you're only getting a couple of offspring per generation, that makes the work pretty tough. So this sped up his ability to collect data. And so what was he looking at? He looked at 7 different traits. You're probably familiar with most or all of these. Usually when people are talking about these experiments, they focus on the shape and color of the seeds and or the flower color, but he was looking at these 7 different characteristics or traits that pea plants have that were really easy to observe. So he could really easily see, from one generation to another, what was happening from parents to offspring. So I'm not going to go over all of flower anatomy. That's not what this is about today. I just added on this word because there's a lot of confusion. In fact, a few years ago, I was looking at some pretty established and scientific text that actually called the pollen grain gametes, but gametes are in the pollen. And likewise over here, the ovules are not the gametes. The gametes are in the ovule. And again, like I'm going to bring it back to the whole evolution idea, which is, if you think about what happened and what's the reason for all of this, this is a lot of packaging, right? This is triple pack. I mean, you've got, a lot, you've got, all of this protection, one layer after another, right? And what's being carried is these gametes that hold this genetic data that is the most important thing for this plant, is to make sure this stays healthy and viable and gets to its destination in order for this plant to be able to reproduce. So, you know, over time, all of this layering has happened, which is a far reach from you know, gymnosperms and even more basic things that came before them, right? So now we're going to dip a little bit deeper and look at Mendel's laws of hereditary. Mendel founded what we know today as modern genetics. We're going to look at these three laws: the law of segregation, the law of dominance, and the law of independent assortment. And so first, the law of segregation. This is an easy one. Sometimes, since all three of them or a couple of them are often used together, and it's hard to figure out what law you're talking about, or that maybe they're all one law, but they're actually all three separate concepts that he posited. So the law of segregation, really easy, is just saying that each parent, so the paternal chromosome, the maternal chromosome, each parent is only passing on one allele for each offspring. And so the parent had two, oh my goodness. So the parent had two chromosomes, right? And then they're passing one of those off to their offspring, and so that's what's happening with both of them. And when these two match up, from the alleles and the chromosomes from each parent, you're ending up with genetic diversity, which is really important for adaptation.
Thinking in the right direction.
Evolution. Yeah.
Hold on. Jerry, can you hear me? Can you mute yourself, please? Thank you. Hold on, Lisa. There we go. Thank you. All right.
Oh, and so then I did add, as you can see down here, because of that confusion with what gametes are. So when people are talking about gametes, so these chromosomes are in the gametes, right? So that's what's being passed on right there. So that's where the traits are. That's the genetic material in there. So that one's easy, really a cinch. Just that, you know, the parents, like each parent has two chromosomes. They're separating. The chromosomes are separating out, and the offspring are getting one from each parent. That's the law of segregation, right? They're segregating. So now the law of dominance there's a little bit more to this one. So in Mendel's initial experiment, he was looking, the P generation, or the parents, the first generation, they were homozygous for one trait, which means that the only genetic material that this parent, the parent on the left, could pass on was that it is, in this case, because Mendel's first experiments were only looking at one trait. So here we're saying this parent on the left could only pass on the color yellow, and the parent on the right could only pass on the color green. So what happened in the next generation? And this is where Mendel started developing his law of dominance. He started seeing what was going to happen in that next generation. And he saw, whenever he did this cross, all of the offspring were yellow. So this is where he came up with his theory, the law of dominance. He established this yellow is dominant. It's a dominant trait, because every time I do this, all of the offspring are yellow. Then what happens in the next generation? Well, in the next generation, now a green pops up, right? So now things, things get a little more complicated, maybe, except that Mendel understood he was a mathematician and a physicist. He didn't get into plants until he was about 30. He was a really strong mathematician and physicist. And so he already had some predictions, and he kind of thought this was going to happen, and we'll see why he thought this would happen in a minute. So here, this is actually the same thing from the last slide, except it's showing you what the alleles are, right? So again, this is the repeat from before. This was the yellow parent, and this was the green parent. The green parent could only pass on what were the recessive green genes, and the yellow parent, the dominant yellow. So all of the offspring got one allele, because the only thing that this parent could pass on were big Ys, right? The dominant gene. The only thing this parent could pass on were the recessive genes. So if it didn't matter which chromosome, went down the line, both sides were going to always have the big Y for the yellow one, and both sides we're always going to have the green. So whichever chromosome went to the next generation during the segregation that we saw in previously, this was always going to be the outcome. All of the offspring were going to have a big Y and a little y. They were all going to be heterozygous. Which means they, versus the homozygous of having both dominant or both recessive. Now, Mendel had worked this out, and he knew that this would happen. Or he saw that this would happen. He started to understand that this would happen. And then he got to a point where he could predict, right? And so this is the famous Punnett square, right? Where you have now this is what the offspring, the gametes of these offspring of this generation. Now you're in the next generation going on, and what happens when they self-pollinate. So we know that this is going to be the genotype of some of them, and this is going to be the genotype, right, of some of them. In other words, all of them are going to have the same genotype. And I say some of them because in other cases it'll start to get more complicated. And so if you do these crosses, what do you end up with? And then once you start to see this at the, what ends up being more the genetic chromosomal level, which Mendel didn't have the ability to see, he didn't have, the technology at that time, you can start to predict. And once he got to the point that he could he started to see this pattern, the pattern of inheritance, then he could predict, right? So for him, this law of dominance gave him the ability, at least with the experiments that he was doing, to see that phenotype, in other words, the way the different characteristics that he saw in the plant would reveal the genotype, the genetics of those plants. Now, it does get a little more complicated, as we'll see in a second, but this law is completely valid for the experiments that Mendel did. Now, this is where it gets a little bit more complicated, is that it just kind of by a fluke, also because he chose a model organism, right, that was very simple, and he had to because he didn't have a lot of technology. Mendel's pea plants had really clear, distinct dominant and recessive traits. So always if the dominant gene was there from either of those parents or both, then you always saw that dominant trait. But if it was only the recessive genes, that's the only time that you would see the recessive trait. Also, and we'll talk about this in a minute as well, there was only one single gene associated with the two allele types. So the only one gene that said you're going to get a yellow pea or a green gene, a green pea, right? So there was only one gene to dictate that color. However, as we know, it's a little more complicated. There are a lot of cases where there are multiple genes, right? But here's we're going to get into. We're not going to get into this. I'm just going to go over this. No, just to make sense of all of the exceptions that we see to Mendel's law of dominance. So there's intermediate phenotype, which is fun for if any of you are doing plant breeding, if finding those intermediate phenotypes where actually, no, there isn't one clear dominant and recessive phenotype that you're going to see necessarily, or genotype, because in the phenotype, the way it looks, there can be an intermediate. So in this case on the right, you've got these snapdragons, and if the R was dominant, then we would have 3 red flowers, right, and 1 pink. However, there's incomplete dominance, or maybe even like co-dominance, right? They share. Neither one of them is more of a heavyweight, right? So the white shows up and the red shows up, and what do you get? You get pink. However, besides that, Mendel's predictions like work. So they still work, knowing that this is incomplete dominance. Now we can still make the same sort of predictions using Mendel's methodology, right? Now we know that if one of them has a big R and a W, it's red and white, then it's going to be pink. So knowing that, we can still use his methodology to make predictions. Then you also have over dominance, which is fun, right? So you're getting, all of this show up in this hybrid and in this monkey flower. So again, this is an extension. It's often called an extension to Mendel's law of dominance because that law is completely valid still. It happened to be that the plants he was working with didn't have, any of these, over dominance or any incomplete dominance or anything like that. And then the last two I'm going to talk about here, extensions, both using ABO blood type in humans as an example, but it's seen in the plant world. But this is an easy example for us to understand. Co-dominance is when two alleles are expressed at the same time. So there's ABO blood type, and A and B are both, the co-dominant. And then with the blood, a distinction between what's happening here versus with Mendel's pea plants is Mendel's pea plants, as I said a couple of times, there was like the peas could either be yellow or green. That was it. There was no other allele that could be passed on. It was either Y or G. Well, here you've got one third possible allele. Now each offspring can only get one allele from each parent, but it's out of 3 options, right? 3 possibilities. So then you end up with a little bit of a more complex Punnett square. And again, this is not to take away from the truth of Mendel's law of dominance. This is just kind of added on because he had no way to know that this could happen, because it didn't exist in his model plants. So now we're going to talk for a few minutes about the law of independent assortment. And if you're still with me, this is where we're halfway through, or maybe even more, the nitty gritty of this. And then it'll be just fun pictures. So the law of independent assortment is saying, so all of the experiments that we were talking about earlier, we were looking at monohybrid crosses. If you remember that first P cross slide that I showed you, we were looking at either the yellow pea or the green pea. And now you're getting into looking at yellow wrinkled pea and green unwrinkled pea, right? So you're looking at two different traits. You're looking at color and whether it's wrinkled or smooth, right? And so what Mendel found was that this happens independently. This sorting and passing on of traits, that each of the traits, those 7 traits that he was looking at, he was never going to end up with offspring that always had all the same traits as you know, that would have come from one parent. It was a mix of what the parent could have given them. Like maybe a parent was, had yellow peas and white flowers, but but they were dominant in one of the traits and recessive in the other, and they could pass on a different mix of those genes. So if this was A and little a, and B and little b, and they were all on the same gene, then that the way these were passed on might differ. Like this big A might be passed on with the little b, or it might be passed on with the big B. It could be a different mix. This is Mendel's work showing those crosses. And so you can see this was before Punnett squares existed. That wasn't until 1905. And so this was his kind of Punnett squares that of sorts that he created. And this is what this looks like. It's probably better to see the pictures than what I was trying to explain 2 slides ago, where you have these two different you're doing dihybrid. So you're looking at the traits of the yellow and round and the green and wrinkly peas and what happens. And now you see that you can get a real mix of, an assortment of different phenotypes here, because look at all of these different genotypes. They can mix up in different ways. And so that's all that law is. And then you can see how this is just another picture of basically the same thing, where you're showing it expanded. So if you're looking at a population, and if you're doing crosses from one generation to another, these are the 4 possibilities. So you see them. These are the only 4 possibilities that you'll see for if you're doing a dihybrid cross like this, and with only two possible alleles, right? And so the mother can only give these, and the father can only possibly give these. So what would happen with the offspring? If you can see this big assortment, what that means is that all of the yellow smooth, if the assortment didn't happen, then it would be always yellow and smooth, or maybe green and wrinkly, or green and smooth, or something like that. There would only be 2 options, as there were before. But now there are more options, because each of the 2 traits mixes independently. So again, you have an extension here. And the obvious example is that there are linked genes. For example, if these 2 genes are actually on the same segment, this is depicting two separate genes. So what Mendel was looking at was like the pea color is not maybe on the same gene as the pea shape. And so that's why they get passed on in a different way. Whereas here, if these genes are linked, it's very easy for them to just travel together when they move on to the offspring. And so you can see here why this worked so well, why this was such a great model organism. Pea plants have 7 chromosomes, and it turned out that Mendel chose traits that were all on different chromosomes. So there was no way that there's no way he could have this would have happened in the characteristics he was looking at. He wasn't looking at any characteristics where this would have been right here on the same gene, right? And so now I'm moving kind of into the modern, right? The modern phase of, I did past and present. So the present now, where we can see these things, and now we're like, oh, this is why Mendel got what he got, which was absolutely right for what he was studying. And I'm not going to read this or go into this, but it goes way beyond this. Plants differently from animals, they can really easily double or quadruple their chromosome number. It happens a lot. This is how a lot of new species have evolved over time, is that instead of just passing on one set of chromosomes, somehow 2 get passed on. And now you can have double the chromosomes in the next generation, or have a triploid. And so it makes for a very interesting hybridization naturally. And then it also makes, so this right here is a triploid, right? Can't see it, but this is what we're looking at, with which and it happens quite frequently in naturally. And in fact, Ruth Kobayashi works with that in some of the plant breeding that work that she does. And this is why you so often see plant hybridization happening. It's much easier to do than with animals. That kind of thing is not so easy with animals. So whether or not humans are influencing the genetics of organisms through hybridization, it's always been happening, as I said before. It's been the origin of species, for example, as we just spoke about in the last slide. If especially with plants, if they're duplicating and ending up with extra chromosomes, you're from one generation to another. There are a few animal species in the animal kingdom that do this readily as well. But plants do it a lot more. Species mergers happen sometimes. And this is, this all happens naturally, and this allows for adaptation. It's really important that especially now with climate change and everything, and there have been a lot of things historically, right, through the eras where, and this is how angiosperms came to be in the first place. It was a very important adaptation to have fruit and to not have a naked body like gymnosperms do. So genetics and hybridization, kind of understanding hybridization, I think it really helps to know a little bit about the underlying genetics. You don't need to, though, in order to hybridize and to breed plants. And so just to finish up, I'm going to talk just a tiny bit about this, somewhere that everyone here, I'm sure, is really familiar with. So for 7 years, I worked on this program from its initiation. And we were breeding plants we're starting with this picture because you can see how from far away, you always see these bands. And I'm sure most, if not all of you, have been to the flower fields and noted that when you get up close, well, geez, really, like I can see what those colors are like by the, the stripes, but when I'm up close, wait, which which color am I in? So this is why we started this program, because we're like, geez, what's happening? Because we've got gold, we've got yellow, we've got red, so this is pink, this is gold, this is orange, this is purple, and there's just an array of colors, and not all of them are exactly the right purple or the right gold, or, and it's not just the flower fields that these plants are grown for. The ranunculus, Milano and Company, who breed these plants, they're in the the cut flower industry, right? So if a wedding planner wants to order, several dozen yellow ranunculus, they don't want to have like 6 gold ones in some of them, and a few red ones and a few that have or 2 colors, or whatever. So we're basically brought in to help clean things up a little bit, and that project again is ongoing, and there are some other experiments that are involved with that. But so this is we're not working at the genetic level, we're not working with microscopes or anything like that, we're just working at the phenotype level. And so just really briefly, the background of ranunculus: this is the region where they come from historically. They're grown a lot, pretty much anywhere that has weather that fits this weather. In fact, it's interesting how successful they are here, because they need a cold season in order to grow well. So that's actually one form of breeding and adaptation that's already has happened in the many years that ranunculus have been bred here in San Diego, because we really don't have much of a cold season, at least not as cold as they would like. And so that's part of the breeding that has already been happening for decades and decades here, because it's actually fascinating that they do so well here. And so this is what you can get. Again, like, if a floral, florist or a wedding designer is, wanting to order, bunches of a certain color or type, they want to make sure that this, like the gold, is like all the flowers in the gold are gold, right? So tools of the trade, if you're doing breeding, I'm not going to be talking about this breeding per se, but these are really the tools that we use. This was it. We used our eyes and our feet and measuring sticks to transfer the pollen. We use tweezers. We use magnifying glasses, mostly because it was fun to get and I really suggest this: if you if you have a magnifying glass, go out in your garden and look at your flowers. It's just amazing. It's really neat to see. And then we do a lot of tallying. Some of our plants are in different experiments, and we do self breeds and cross breeds, and so sometimes we cut and tally. Sometimes we also need bags to, when we pollinate, we want to bag those plants to make sure that they don't also get pollinated by something else. And then we have running records of what all of these crosses of generation after generation, akin to what Mendel was doing. And then last, that we use, that you probably won't use in your garden, is that we defined the colors that we were using. And so we actually had used Pantone colors, and we chose maybe 3 or 4 different colors that were really close, and we decided this is going to be rose, and the same thing for gold, and the same thing for yellow. And so We have a volunteer program, and all the volunteers and we, the staff, would go out, and we have a set of these of examples of the Pantone chips that were appropriate for each color. So when we weren't so sure, we could put the flower right next to the gold. And if it was gold indeed, and if we were looking for gold, we were in a gold section, then we would flag that plant, and that would be part of what we'd make sure we'd save for the next year. By the way, there's a mother block at the flower field. So right now, I think it's well, for the last couple of years, it has been the most northern field there. And so no cutting is done there. So all the flowers that are used as cut flowers for the flower fields are taken from all the other fields. But obviously we don't want to lose the genetic material that we're really looking for, the really prime, perfect flowers. And then, so just really basic, you all probably know there are two ways to self-pollinate, and one flower can pollinate itself or another flower on its same plant because it's going to have the same genetic material. It's a self-pollination. And then one way to have cross-pollination, right, going where the pollen goes to another plant. This I wanted to show real quick, just because right here, this is the pollen dehisced. So when it's not until it looks like this that it's ready to be transferred. And you can see that this guy's got a bunch on him. Before that, when you look at the anthers, they'll be like shiny. And then this is part of the cool part if you have a magnifying glass to look at. So now I'm just going to fly through a bunch of different pictures to show you the variability. And aside from color, what we're looking for out there is kind of like Mendel had his list of characteristics, we did too. So it had to be the right color. It had to have the right number of petals. We didn't count the petals, but we called it double, which just had to be really fat, basically. And sometimes it's deceiving. Like this one looks like a double, and then you'd go squeeze it and it was all air inside. And so that wouldn't be a double, maybe. It also had to have the right shape, and this is probably the closest to the right shape that I see here, even though this wouldn't be a full double. And so basically none of these would pass the test, none of these here on this plant. But look at this. So, and you've got more than one plant here, actually. So this is probably one plant. This is another. This is definitely another, right? This color is called flame. And then you see here, this is more like what a ranunculus looked like originally. It still has more petals than it used to. But this is this you can see on the same plant. This would be a single, and this one right next to it, if it was on the same plant, and I think it was, it would have probably been a double. So that was another thing that we looked at in terms of characteristics, is that a really good plant had to be a certain height, had to be the right color, had to be a double, had to actually have at least 3 doubles too. And sometimes that was the clincher. Sometimes there were just too many. There was one double and the rest were singles. This is really gorgeous. One of the other things, though, one of the other criteria is that the plant needs to have seeds. We need to pass on the genetic material. And I'm not opening this one up right now, but I could almost bet you this one probably doesn't. It doesn't. And I'll talk about that more in just a second, although we're going to fly through this. So again, look at this variability. This beautiful, but yeah, sorry. This is really awesome. You can see right here, right? No seeds. What happened? Again, adaptation. Those seeds, the plant, we've been breeding and breeding and breeding. It's like you only have so much energy. The plants only have so much energy. If you remember way back here, look at all those seeds, only a few petals. So I'm kind of giving away what happened here.
Yeah.
The seed, they became petals, right? And you're going to see it's a little more obvious in a second. So, you know, there's always a trade-off. And what we're doing, we're not working in a lab like, you know, under a microscope, being able to transfer genetic material. We need those seeds in order to do our work. Again, really cool looking. You can see something funky going on. You've got some anthers sticking out right here, right? This has so many petals. This might not even have any anthers on the inside. It's got some anthers sticking out. So this is what happens when you're hybridizing, right? And again, nature does it by itself too. But when we're trying to force more petals. And so we're seeing all of this funky stuff. This is the one I was talking about. So you can see this even retained some of that green color, and you'll see this fairly often out there. This yellowing, I think, is just from age. It might not be, because it looks very much like that green. But this happens a lot, and these are, again, the anthers. Sometimes it's really obvious, and you can see how really funny these petals are getting on the inside. The plant doesn't know what to do. I think this one's gorgeous. Again, you know, it's got these little frilly things, which are totally unacceptable for our program, but gorgeous. Really cool again. And again, you're seeing seeds disappearing. I bet you there's none there. There you got a bunch, right? But not very many petals. Really cool color. Yeah, right?
Wow.
This one's got some weird things going on here. No seeds, no genetic material from this plant, which is really a shame because this is our color salmon, and we've been having a really tough time with that color. The first year we did this program, we used to see like dinner plate size, like flowers, salmon flowers. They were just so gorgeous, but it's been really having a tough time. We have trouble getting genetic material. This I bet was in the salmon as well. It's too pinkish. But it's not just the flowers. Even after the flowers are gone, there's uniqueness, right? And so with that, I don't know if you know, but James Hubble just died recently, and I saw this beautiful quote from him. The interconnection with all life is not a random thought. To me, it is a fact. And so I hope for you, as for me, all of your plant-related journeys are people journeys as well. It's really wonderful to make connections with other people through gardening, through horticulture. And so that's been just as much a gift as the plants themselves. And so just want to thank you for inviting me. I hope everyone learned something and
Yes.
at least enjoyed the pictures, if not.
Let's have you stop sharing, and then I'll try to do some questions.
Yeah.
Thank you. Okay. So, there you are. Any questions in the room? I'm looking for chat questions right now. So I think I learned something about my own family.
Oh,
And so my dad was number 9 out of 12 kids.
Ooh.
His mom and his dad's sister and his mom's brother married and had 2 kids. So I have double cousins.
oh.
And
Yeah.
it looked like those peas. I was like, oh,
Yes.
Aunt Trudy.
Yeah.
But that I think I had an epiphany about my own family from what you said. So thank you. It was
Oh, awesome.
because
I
it was
love
a very,
that.
it's just always knew that there was this, big mishmash in the gene pool, and it was, similar but different.
Yeah,
Anyway,
that's great.
yeah, so I'm a P. I'm a P. Th- what about in my yard, I have scented geraniums that I planted, I'm going to say 20 years ago or more. And they were different varieties, 30 at one time in my yard. And now I think I have 5. They all did their thing over time, and they're not really what they started out as. And so I noticed your bands at the flower fields. I tried to do bands 24 years ago as an idiot, but anyway, I had my each one in little swales along a really large area, and they didn't stay. I mean, it was a hot minute and it was all together.
Yeah.
So your bands of things, how is that maintained?
Well, those bands aren't separating them for breeding purposes. That's
Oh,
for the public. What we're doing is doing the separation. For example, with sometimes we use windrows, but in our case, well, we did that the first and maybe second year. But in our case, what we're doing is when we pollinate, when we find 2 golds that are pristine, we're like, these are both great, we want these 2, we're playing matchmaker, right? We want babies from these 2 parents. So we pollinate, we take the pollen from one and pollinate the other plant, because we want genetic diversity, right? But we still want to maintain the look that both of these plants have. And we bag them. That's why we bag them. That's how we prevent that cross pollination, because even though we have the bands, their ranunculus are wind pollinated. They're not insect pollinated. So they're free to go wherever they want to go. Yeah.
okay.
Yeah. So
Fascinating.
bagging, I would say absolutely bagging. It's the easiest thing.
I'm not bagging mine, so they're gonna just
Yeah.
keep morphing into who knows what.
Yeah, well, that's fun too.
Yeah,
I
no,
love
it is.
that.
It is.
Yeah,
I've made my peace with it. They taste
Yeah.
good. That's all that I really care
yeah,
about.
agreed.
But the plan of it originally was completely different than how it's turned out. So
Yeah, no, this isn't about, that keeping things the same all the time is great or anything like that. I love the diversity too. I think understanding genetics is just interesting because you understand what's happening under that. That doesn't mean you need to use that knowledge, that information to change things though.
sure. Susie.
Yeah, I have a question. Do you think Gregor Mendel lucked out with peas, or do you think he had a feeling going in that there was going to be a relatively simple inheritance?
The question is, do you think that Mendel lucked out with the peas, or what was the rest of it?
Do you think he had an inkling going in?
Oh, did he have an inkling going? Oh, got it. Did he have an inkling going in that this is what he was gonna find?
I have wondered that myself. The one
Oh.
thing I will say though, and then maybe there's something out there that says that, and of course I'm gonna have to look it up after this. But the one thing I'll say is that he was choosing a model organism. So it had to be really simple. It had to be, he had already seen, what pea plants were doing, and he knew that there were only 2 colors for the flower. He knew there were only 2 colors. So it was given that that was all simple. There's no, that it was going to be simple to do those crosses, right? That's why it was a model organism. However, really, he couldn't have known initially, by initially, I mean before, I mean, maybe he knew before he started experimenting, but it would have had to be from observation that each of those genes were on, that each of those alleles were on different genes, on different chromosomes. There's no way he could have known that because he couldn't see chromosomes. He didn't know what chromosomes were, but he might have known the phenotypic result of that. He might have realized before he did the serious experimenting that the traits were not always carried together, right? That they were all separate. He might have known. I wouldn't say that wasn't possible. But either way, it would have been through, whether it was from his scientific observation or from previous just day-to-day observations, he realized that this, you know, that wasn't luck because of that. Yeah. But choosing a really simple model organism by default sort of might more easily lead to this situation.
So interesting. Anybody else? This is deep. This is deep. This is, yes.
I'm just wondering if they do the Mendel system for watermelons, like now we have seedless watermelons, was that developed from that?
The question is watermelons and seedless watermelons, is this the same sort of?
Same thing, like you see, that's the same. Yes, you can look across any sort of breeding. That the example from the pictures I was showing you would be the petals, right? So we've got, so seedless, it's exactly the same thing. You're getting more, the plant has to give something up, right? It's giving up the seeds to just give you fruit, more fruit in the place where the seeds would have been, you know?
Sure. Yes.
How do you propagate it if it has no seeds?
How do you propagate it if it has no seeds?
Well, we're not doing that in this project. There you're going into a lab, right? But we specifically didn't want to do that because you can use, you know, different genetic material and different plants, as you know, some you can propagate through pups and things like that. So there you're getting into a lot of different possibilities because some plants are going to have certain traits that they're going to be more inclined to pass on or not pass on. Some plants will not self-breed, so you can't do self-pollinations. They'll only do cross-pollinations or only do self-pollinations under dire circumstances. So there you're getting to a species by species examination where you're going to have to look at what does this species tend to do.
Yeah. Yeah. Wonderful. Thank you so much.
Yeah, thank you. I warned you that it was deep. I was
Yeah. No. I took notes and I don't know that I'm going to understand them. I had 3 years of German, by the way, in high school, and I couldn't read that Mendel. So yeah. Oh, one more
quick question.
What?
Who is sponsoring this project that you're working on?
Who is sponsoring this project?
There isn't a sponsor. I was an employee of Milano and Company. So I had an employer. So Milano and Company, their main growing fields are up in San Luis Rey, and where they grow tons of different species. They've been the #1 provider for the Rose Parade for years and years and years. They grow, they import, they export. They do a lot. They now do flower arranging as well. And then they have the ranunculus at the flower fields, and there are a few other things that are grown there: coffee, blueberries, and there's some UCANR experiments going on there as well. But yeah, Milano and Company. So we actually have a lot of ranunculus over in San Luis Rey as well. And so some of our work is up there as well, because ranunculus, so they can propagate by, see, this is a whole other thing. They can propagate. This is why you have to look at each individual species by seed or by tuber. So somebody asked what happens if there's no seed. So we can still use the tubers, but but what we do is alternate. So one generation we're planting the tubers, the next generation the seeds. Then I'll keep alternating, and we find that that works better than planting just the seeds or just the tubers. However, you still need that alternate season, the seeds. So we still want plants that are producing seeds. So we're not, they're much more hardy if you alternate like that. So we wouldn't necessarily be in a bind with the ranunculus if they didn't produce any seeds, but we'd lose some of the vigor.
That's so fascinating.
Yeah.
Oh my gosh. Wow. Okay. I appreciate you greatly. Just for everyone, this was recorded. It will take a while, but it will end up on our YouTube channel, so that you didn't really have to take notes if you wanted to watch it and take it over again. That'll happen. I used to be able to do it in a couple of weeks. Now I can't. It takes me a little longer. The process is just a little longer. So give me a little bit more than a couple of weeks to get it up online. All right, Lisa, thank you so much.
Thank you. Thank
Yes,
you
I hope
all.
you can hear. I hope you can hear.
Yeah.
All right.
Thank you.
Thank you. Talk
Take
to you
care.
later.
Bye-bye.
Bye. Bye, everybody in Zoom land.